It is so cold in Minnesota right now. So cold, in fact, that my fingers and toes are freezing up. Did you know that people actually have an extreme version of this where they completely lose circulation in their fingers and toes? It's called Raynaud syndrome. Actually, scientists recently discovered that Raynaud syndrome is likely a genetic disorder, which means that we could probably treat it with CRISPR-Cas9 technology. Now, you might be asking yourself, Kiko, what is CRISPR-Cas9 technology? I'm really cold, so why don't we go inside, grab a warm mug of hot cocoa, and I'll explain it some more.
Okay, now that I've warmed up and gotten some cocoa, I can tell you more about CRISPR-Cas9. CRISPR-Cas9 is a genome editing tool that can change specific parts of DNA sequences. CRISPR itself stands for Clustered Interspaced Short Palindromic Repeats, which Dr. Stanley Chi, a bioengineer from Stanford University, recently explained is like a bacteria's immune system. When viruses inject and infect bacteria with their DNA, CRISPR makes a sort of guide to identify that viral DNA in the future. Bacteria are then able to use Cas9 to attack future infections. Cas9 is a protein within the CRISPR system that can locate injected viral DNA and cut it, degrading it until it's no longer harmful. So how does this bacterial process relate to human cells? Dr. Jennifer Doudna, who discovered CRISPR-Cas9, found it can be programmed into human genomes too, sending Cas9 to specific DNA sequences in human genomes and cutting the DNA, thereby inhibiting that cell from making harmful disease-causing proteins. Cells are really cool because they can actually detect when a part of its DNA is broken and then quickly fix it. So, when a mutated DNA sequence is broken by Cas9, the cells will repair it with a proper sequence that no longer codes for harmful proteins. By cutting out these genetic mutations, scientists can use CRISPR-Cas9 to treat genetic disorders. In fact, it's already being done. In the last couple of years, the FDA approved the first drug that uses CRISPR-Cas9 technology, Casgevy. As explained by the FDA, Casgevy is able to modify the genomes of blood stem cells to treat those with sickle cell disease. Now, you might be finding yourself asking, why aren't scientists using it to treat genetic diseases all the time? Well, Dr. Doudna has reported that CRISPR-Cas9 unfortunately raises some ethical concerns surrounding its testing often being done on embryos and that it could enhance physical traits, like giving people different eye colors and stronger bones. Because CRISPR technology is still fairly new, clinical testing has been slightly slowed down until more information can be gathered and more regulations put in place on CRISPR-Cas9 technology. However, there remains a lot of optimism for advancements in areas like how to deliver CRISPR into cells, how to control DNA repairs and unintended cuts to DNA, and overall research on developing treatments for thousands of genetic disorders using CRISPR-Cas9 technology. So who knows, maybe in future years we'll have a cure to Raynaud syndrome after all.